Okay, so welcome to this next video in which we are studying the selectins. Okay, so we discussed in the previous video how the selectins are a family of cell adhesion molecules. Okay, now it's a small family, a very small family compared to the integrins and the immunoglobulin superfamily cell adhesion molecules. Okay, it only contains 3 members, and these are P-selectin, okay, then E-selectin, and finally L-selectin. Now there is logic in these names. P-selectin was named P-selectin because it is found on platelets, although that isn't the example we will use. E-selectin was named E-selectin because it was found on endothelial cells, and L-selectin was named L-selectin because it was found on lymphocytes. Okay, so let's start with studying P-selectin. Okay, so P-selectin is used on endothelial cells to help the recruitment of neutrophils. Okay, so let me show this. So if you've got an endothelial cell here that is in an area where an infection is occurring, so if you've got an infection occurring in a certain portion of tissue in the body, then the endothelial cells there will undergo type 1 activation. Okay, they'll also undergo type 2 activation, but it's type 1 activation that will result in P-selectin being put on their surface. Okay, so the endothelial cells and the capillaries and the post-capillary venules will get P-selectin on their surface, and this is going to help them with the recruitment of neutrophils. Okay, so here is our endothelial cell here, sitting on the basement membrane, and it's going to end up with P-selectin on its surface here. Okay, and P-selectin has a ligand. It will bind to a molecule on the surface of neutrophils. So let me draw a neutrophil here. Okay, so this is a neutrophil. Right, so neutrophils have multi-lobed nuclei. Okay, they don't have multiple nuclei, but they have a single nucleus, but it's got a very strange shape, basically. It's got this lobar shape, where it's got lobes and then little tubes connecting the different lobes. And the number of lobes that a neutrophil nucleus will have is not set, basically. So if you look at different neutrophils, you'll find a different number of lobes. Okay, but the archetypal number of lobes for a neutrophil nucleus to have is usually around 3. Okay, so this is our neutrophil. Now, neutrophils are a phagocyte. Okay, so if we bring them into the site of infection, they'll start engulfing the pathogen and hopefully breaking it down and destroying it. Okay, so they are part of the innate immune system. They can attack any pathogen and they'll do it in the same way. They won't do anything specific for different types of pathogen. Okay, now usually they don't stick to endothelial cells, and this is because the endothelial cells usually do not have P-selectin on their surface. The neutrophils, however, always have the ligand for P-selectin on their surface, and the ligand for P-selectin is what's known as P-selectin glycoprotein ligand 1. Okay, so this is P-selectin glycoprotein ligand 1, and that's often abbreviated to PSGL1. Okay, so for short, PS for P-selectin, G for glycoprotein, L for ligand, and then 1. Okay, so let me colour these in separate colours. So P-selectin glycoprotein ligand 1 is here in blue. Okay, P-selectin will have in purple here. Okay, and the two are binding together, and that's binding this neutrophil to the endothelial cell. Now let me stress, this would not usually happen because the endothelial cells wouldn't usually have P-selectin on their surface. So it's only going to happen in the capillaries and the post-capillary venules of an inflamed tissue, because only those endothelial cells will have been activated to put P-selectin on their surface, and it's type 1 activation of endothelial cells which results in P-selectin on the surface of the endothelial cell. Okay, now this will help in the adhesion of the endothelial cells to, the neutrophils to the endothelial cells. In fact, it's what's known as tethering of neutrophils to the endothelium. And this is going to help with the recruitment of the neutrophils to the site of inflammation. So what's next going to happen is this neutrophil will diapedese across the endothelium, and it will end up in the interstitial fluid where you have the infection, and then it can go and hopefully attack the infecting pathogen, the invading pathogen, and help to clear the infection. Okay, so that's P-selectin. Now, I want to stress that P-selectin glycoprotein ligand 1 is not a cell adhesion molecule, okay? And this is an example of where P-selectin is binding to something that's not a cell adhesion molecule, even though it is a cell-cell interaction. It's not an interaction between two cell adhesion molecules, so this is not a cell adhesion molecule. And we'll see another example of this with E-selectin and also L-selectin as well. So let's have a look at this then now. Okay, so what it really emphasizes is that these rules up here, they're guidelines, but they're not strict rules, to use that expression from Pirates of the Caribbean. Okay, so they're more what you would call guidelines than rules. Right, okay, so let's have a look at another one now. So here, again, E-selectin is put on the surface of endothelial cells, which are lining blood vessels, capillaries and postcapillary venules in sites of infection. Okay, now type 2 activation of endothelial cells will result in the endothelial cells putting E-selectin on their surface. So here is E-selectin on the surface of this endothelial cell. Okay, now type 2 activation takes slightly longer than type 1 activation. Type 1 activation is induced by things like histamine and occurs within minutes. Type 2 activation is induced by things like interleukin-1, tumor necrosis factor alpha, and it takes hours generally. Okay, because it involves changes in gene expression. So here comes E-selectin, and we've now put it on the surface of the endothelial cell. So here in red is our E-selectin on the surface of our type 2 activated endothelial cell. And again, it's going to bind to a ligand which is always on the surface of the neutrophil. So the reason the neutrophils don't usually adhere to the endothelium isn't that they're changing their ligands, it's that the receptor for the ligand on the surface of the neutrophil is not usually present on the endothelial cell. Okay, so here's our neutrophil with its multilobe nucleus, and the molecule that it's going to bind to is known as sialyl Lewis X. Okay, now this is a very small little molecule that you can stick onto the side of proteins and other things that are on the surface of the endothelial cell, and this is what E-selectin will be binding to. Okay, so let me colour it in orange. So this is sialyl Lewis X. So again, it's not actually even a protein. So it's certainly not a cell adhesion molecule. So again, it's breaking the rules. Sialyl Lewis X is more like this sort of interaction where you've got a cell adhesion molecule binding to the extracellular matrix. However, obviously this is between two cells, so these rules that we came up with before, they're guidelines. Okay, so this is the role of E-selectin in the immune system, and again it tethers the endothelial cell to the neutrophil, or rather the neutrophil to the endothelial cell, and the neutrophil will then diapedese across the endothelium into the interstitial space, and it can then go and fight the invading pathogen. Okay, so let's finally turn our attention to L-selectin. Okay, so L-selectin is involved in the movement of lymphocytes, specifically naive T lymphocytes. Okay, so for this, let me draw a picture of the lymph nodes. Okay, because we need to understand something about T lymphocytes. So, which are naive T lymphocytes. So we're not talking about ones that have been activated, we're talking about the T lymphocytes which are sitting in your lymph nodes waiting for an infection, basically, waiting for a pathogen to come along that has their antigen. Okay, so we're not talking about T helper 1 cells, we're not talking about T helper 2 cells or cytotoxic T cells, we're talking about naive T cells of both CD4 positive and CD8 positive types. Okay, so here's our lymph node. Now these small vessels that I've got here, these are the multiple afferent lymph vessels which are coming into our lymph node. Okay, so every lymph node in your body has a huge number of afferent lymph nodes coming into it, which are all draining lymph from the peripheral tissues. So all of these small blood vessels, well, not blood vessels, sorry, they're lymph vessels, are bringing lymph in. Okay, so in comes lymph through these tubes here. Okay, and this final big lymph vessel here, this is the efferent lymph vessel, which means that it's taking lymph out of the lymph node. Okay, so this is the efferent lymph vessel. Now, oh dear, what have I done? Afferent lymph node. This should be an afferent lymph vessel. Okay, and the same for this one here, efferent lymph vessel. So the whole thing is the lymph node. This is the lymph node here. Now, basically, when the afferent lymph vessels bring in their lymph, what will happen is it will go into a space that's around the outside of the lymph node, okay? And this space is what's known as the subcapsular sinus. Okay, so this is the subcapsular sinus because it's underneath the capsule of the lymph node. Okay, now you're bringing in all of this lymph into this subcapsular sinus, and basically it now needs to get to the efferent lymph node, and this is what's ingenious about the structure of lymph nodes. I quite like this. Okay, to get to this efferent lymph node, it has to percolate through loads and loads of lymphocytes, basically naive lymphocytes of both the B-cell lymphocyte and T-cell lymphocyte type. And again, with regards to T lymphocytes, you'll have both CD4 positive T lymphocytes and CD8 positive T lymphocytes. So you have two layers. You have what's known as the cortex. Okay, so this layer here, this is the cortex, and in the cortex you have special structures which are basically spheres of B lymphocytes, okay? And these, obviously, because we're drawing a two-dimensional picture, they'll just look like circles rather than spheres, but in reality they're spheres. And these are known as the B-cell follicles, and these are absolutely full of B lymphocytes, basically, that have never seen antigen, so they're naive B lymphocytes. Now the rest of the cortex that surrounds the B-cell follicles, so these circles, these are the B-cell follicles, so these are where the B cells are living, okay? But surrounding the B-cell follicle, the rest of the cortex is made up of T cells. Okay, so all of this surrounding space that I'm colouring in blue, this is all T-cell territory. Okay, and you'll have both CD4 positive T cells and CD8 positive T cells in here. And again, they're all naive T cells. They have never seen antigen. Okay, and then you have the next area down here, which is again T cell territory. Okay, now this doesn't have B cell follicles in, is the difference. And this is known as the paracortex. Okay, so again, it's a great big store of T cells. Naive CD4 positive and CD8 positive T cells. Then you have this final sinus in the center here now, known as the medullary sinus. Okay, so let me label that up. So this is what's known as the medullary sinus. Okay, so the whole structure then now, what happens is that the afferent lymph vessels drain lymph from the peripheral tissues into the subcapsular sinus. Okay, the lymph then has to percolate through the cortex and the paracortex of the lymph node to get down into the medullary sinus, and then has to go out the efferent lymph node. So basically, all of the lymph has to sort of pass by the lymphocytes, and this is what's brilliant, because this lymph will be bringing potentially antigen presenting cells back. It will also potentially be bringing antigens in, and basically all of the lymphocytes are getting exposed to the antigen presenting cells and the antigen as well. Okay, so this is how the lymphocytes can spot if antigens are appearing in the lymph, because the lymph has to pass very close to them to get from the subcapsular sinus to the medullary sinus. Now, you have these great big stores of naive T lymphocytes, but the question is, are they static? Is there a T lymphocyte sitting here that's been sitting here since you were born, just sitting there waiting? Is the life of a T lymphocyte that boring that it sits in the same lymph node all the time? Well, the answer is no. T lymphocytes have somewhat of a more interesting life, at least, than that. So basically, there are blood vessels supplying the lymph node, as with everything else. And in these blood vessels, the post-capillary venules of the microvasculature supplying the lymph node, they're different from normal venules, okay? They're what are known as high endothelial venules. So remember, your blood vessels, high endothelial venules. So remember, the basic structure for blood vessels is that you start off with arterioles, then you go into capillaries, and then venules. Let me draw this out. So the microvasculature works like this. You have an arteriole which will split into multiple different capillaries here, which are smaller, and all of these blood vessels are tiny, which is why they're referred to as the microvasculature. Okay, and then the capillaries will reconverge. Okay, what am I doing here? Yes, like this. Okay, to form a venule, or a post-capillary venule, as it's called, because there are different scales of venule. This is a tiny little venule that just follows the capillaries, so this is a post-capillary venule. The arteriole also would be known as a terminal arteriole, because it's the last arteriole before the capillaries. So this is the terminal arteriole. Okay, so these venules that you have within lymph nodes, they have a different structure to normal venules. Okay, so let me show you how they have a different structure to normal venules. So if we look at the structure of one of these post-capillary venules in lymph nodes, basically they have very tall endothelial cells. So let's draw a tube. That's always a good start when you're drawing a blood vessel. Okay, and the endothelial cells don't look like normal endothelial cells. They're really tall, like so. Okay, normally, let me show you a normal blood vessel. Okay, so this is our high endothelial venule. Okay, our normal blood vessel might look something like this. So you'll have these little flat endothelial cells on the basement membrane. And remember, these blood vessels, these post-capillary venules, have a very simple structure. They literally consist of the endothelial layer sitting on a basement membrane. That's all. They don't have any more complicated layers than that, so they're very simple structures. Okay, and this is the normal structure of post-capillary venule, i.e. you'll just have these flat squamous endothelial cells on the basement membrane, which I've shown here in turquoise. Whereas the high endothelial venules, which you have in lymph nodes, these have these very tall endothelial cells, which is why they're called high endothelial venules, because the endothelial cells are high. Okay, so what's the significance of this? Well, basically, these high endothelial venules are what move naive T lymphocytes from the blood into the lymph node, and also what move T lymphocytes back out of the lymph node and into the blood. So T lymphocytes can move across these high endothelial venules, and they're specifically naive T lymphocytes of both the CD8 positive and CD4 positive type. So they can move from the blood into, and it's a little squashed T lymphocyte here, but never mind. So they can move from the blood into this lymph node, and they can also move from the lymph node back into the blood. So they are mobile, basically, T lymphocytes. They don't sit in one lymph node forever and ever and ever. They can move out of that lymph node, they can go into the blood, and then they can end up in another lymph node or another secondary lymphoid organ. Okay, so lymph nodes are all secondary lymphoid organs, which are where you store naive T lymphocytes. Okay, but there is also the spleen. You have the white pulp within the spleen, and that counts as a secondary lymphoid organ where you can store naive T lymphocytes, so they can move into the white pulp of the spleen as well. So you've got naive T cells which can move freely, basically. They can go into one lymph node, come out of that lymph node, go to another lymph node, potentially go into the spleen. So they do have more interesting lives than just sitting in the same lymph node for all eternity. Okay, and we'll continue this discussion in the next video.